It's both a pleasure and a privilege for me to have this job of interviewing you, and I think we ought to get on with it. And why not start at the beginning, where you grew up, your aspirations, your interests?
Right. Well, I grew up in the western part of the state of Washington in a little town called Aberdeen, which had a grand population of 21,000 people. And then obviously I went to school there, and in going through school, my focus, interestingly, in terms of what I ended up doing, was to go to law school. My father was a lawyer. I ended up in the banking business in Aberdeen during the Depression. And so I went on to the University of Washington, fully thinking that I was going to be a lawyer somewhere along in there. But as I got involved in this, I, of course, had to take some science courses to graduate. And this was in approximately my junior year that my career changed rather dramatically as a result of taking a course in zoology, which was taught by a lady by the name of Dixie Lee Ray, who became quite a national figure, actually. She, subsequent to her work at the University of Washington, she became head of the Atomic Energy Commission, and she then became governor of the state of Washington. But at the time I encountered her in this course, which nowadays would probably be called a biology course, she was a marine biologist giving lectures to 200 and 300 students in an introductory course in biology. And she was just one spectacular lecturer. And I thought this was pretty neat stuff. And I became interested and began to think, well, maybe there was another career other than being a lawyer. So I think she was fairly pivotal in what I ended up doing. So I took additional science courses and stayed in at the University of Washington.
So at that time, did a career in medicine pass your...
Well, it did. Yeah, this was kind of the alternative. My grandfather had been a physician, and I think my mother had periodically hinted that, you know, maybe wouldn't that be interesting to think about being a doctor, but I hadn't noticed up to that point. Yeah, I think she was rather pleased when I changed. I don't think my father really cared one way or the other, as long as I was happy doing what I was doing, which was kind of neat in a way. I think I had very supportive and tolerant parents in all of this. But yeah, I thought it would be interesting. Medicine would be a very interesting career, and so I plunged in to all of this. And it was a rough transition, I must say. It was nothing very smooth about it, because I went from classes in which the primary way of responding was essays to classes in which I had to answer objectively in terms of science, and I was suddenly into this incredibly competitive environment of pre-medical students. And this was a jolt, but somehow or other I survived it.
So when you thought about medicine, what did you see yourself doing in the future? I mean, did you see yourself as a practitioner, or? I
think I must have. You know, as I try to think back on it, I occasionally have wondered, you know, how did I conceptualize this? I certainly had that in mind, and exactly what I was going to do with all this, I don't think it was entirely clear. I was fascinated by the biological issues, and the idea that of practicing medicine was of interest. I mean, our family had a number of physician friends, and these were people that I knew, and I kind of knew what they did, and I thought that was a rather admirable career, actually. Mm-hmm. But I think buried underneath it all, there was this kind of interest in the biological side of it as well. But I hadn't been a scientist, so it was kind of dreaming to think that's what I'd end up doing.
So you got your MD from the University of Washington?
Yeah.
And
after again a jolting beginning. I mean, you know, I think for a social science student, I don't know what students are who come to the Washington University. I know they take some with social science background, but that is a rough introduction when you come into a medical school with largely a social science background. I was scrambling the first year to try to figure out, you know, what all the terms and things meant.
So, when did things begin to happen that it would become clear that you really were interested in the nervous system, and this was a...
Well, that happened actually very early. As you know, at that time, Fred Plum was chairman of neurology in Seattle, and we had this in the first year. I think we were in sometime after the first of the year, we began a course on the nervous system. And each Friday, Fred Plum would come in and talk a little bit about its relationship to clinical medicine. And I was really intrigued by the whole thing. I was just intrigued by the subject matter, and I was kind of captured by his style and the way he talked about the nervous system and all of that. And you know, you tend to remember certain things, but I was rather quiet and shy, and I remember him asking some question of the class about pain, and I was the only one that had my hand up for an answer, and by God, it was right. And it was one of those funny interchanges that really kind of you remember. But I just found that very enjoyable. I remember, you know, the textbook on neuroanatomy, Ranson and Clark. It was, I think, the only textbook in my first year of medicine that I read cover to cover. So I found it very interesting in that first year, and I think that course and Fred Plum was instrumental at that point, and subsequently, of course, he has been very instrumental in my career.
So, when you graduated, did your internship and your graduate training, that was all at the University of Washington? No.
I at that point, you see, in order to get into medical school, I had to take an extra year of science courses, and by the time I got through medical school, I had been at the University of Washington for 9 years. And when I first entered the university, I had given serious thought to going someplace else, and then I ended up there just because it seemed like the right place for me. But I figured 9 years was enough, that I should really go see what the rest of the world looks like. So I initially went to Baltimore, was part of the Hopkins system, and took 2 years of internal medicine, because at that time that was highly recommended for neurological trainees. And then I went on to Cornell and the New York Hospital.
So that's where you picked up Fred Plum again.
Well, yeah, I picked him up, of course. You know, I saw him in the first year, and then in the third year, just as they do here, you have clinical rotations, and there one of the major rotations was from neurology. And it was, again, it just seemed to work right for me. Fred Plum would make these rounds at the King County Hospital every Thursday, and they were very, very formal events. So formal, in fact, that medical students were rehearsed beforehand for their presentations in terms not only of what you said, but where you stood and how you handed him the chart and all of this sort of thing. And somehow or other, it worked well with me. And we hit it off. It was because Fred just struck terror in the hearts of medical students. He was a very formidable fellow, and along with a number of other people on the faculty, but he stood out. And for whatever reason, it just kind of worked right for me. And so we hit it off well. And at the end of my third year, he accepted the chairmanship at Cornell. He had originally been from Cornell. He was a student of Harold Wolff. Mm-hmm. And had been trained in the Cornell system, and Harold Wolff died. And Fred returned to Cornell. And so I went to his office and I said, I'd like to take some elective time. So he said, fine. And so I spent a good bit of my senior year at Cornell, actually, on his service. And then we were required to do a neurosurgical rotation. And so I approached Bronson Ray, who was the neurosurgeon at the time. And he was good enough to say, sure. I gather it turned out I was one of the few medical students who ever rotated on his service. That was not the usual sorts of things. And that, too, was a wonderful experience. I must say he was he was a commanding presence in an operating room. I've held the sucker for Bronson, and I treasure that. I think that was just kind of special. And he sent me a book afterwards with a nice inscription, but he was an impressive man as well, a real gentleman.
So now, didn't you, am I correct about you doing some early work with Fred Plum?
Yeah, through these, actually going back into medical school, when I entered, of course, I entered as a person without a scientific background, and I know a number of my classmates spent summers working in people's laboratories, and my initial attempts to get such jobs were turned down. And I just really was interested in working in the lab, so that side of things was of interest to me. So although I had done some rather modest things up to that point, I was determined when I was a neurology resident that I was going to spend time working in a lab and learn something. So he arranged for me to do that, and I worked with him, actually, he and Jerry Posner, for a period of time.
Yeah,
Now that's how I got introduced to all this issue of measuring brain blood flow and metabolism, all of that, in which we did animal studies as well as studies on ourselves, actually.
Are any of those worth talking about?
Well, it was kind of a, I don't know what it's worth talking about. It was it was kind of an experience. There was this whole idea at the time about, or there were these questions which actually haven't been answered even to this day about what exactly controls brain circulation. It's still kind of an up in the air question. And one of the prevalent idea at the time was that it was the changes in pH around the cerebral vessels that were mediated by CO2 production in the tissue. And there had been this very interesting experiment done at high altitude by John Severinghaus and Tom Hornbein and others in California, which showed that you adapt, your blood flow adapts, and naturally you hyperventilate at altitude. So there's some nice physiology built into all this that allowed one to dissect all of this out. Well, we decided to do a sea-level version of this thing, and we did experiments on animals, and it just seemed to be very complicated, and the answers were ambiguous and unclear. And so at some point the decision was made that we would do this experiment on each other, and the experiment essentially involved instrumenting ourselves with catheters in our carotid arteries and catheters in our jugular bulb. And we laid on a hospital bed and made resting measurements of blood flow. Then we hyperventilated to a CO2 of 20 and held it there for 5 hours. And then we returned to the standard PCO2 at the end of 5 hours. And so Jerry, actually, Posner, did it twice.
I want to be sure I understand. You actually had a carotid puncture?
I had a jugular bulb puncture. They punctured the jugular bulb and they put a catheter in my femoral artery.
Femoral groin.
Yeah, and it stayed in there all day. And the thing that I actually went first, because I was afraid if I saw this experiment on somebody else, I might chicken out. So I thought, I'm not going to do it that way. I'm just going to do it. And Gordon Potts, who was the neuroradiologist in New York Hospital, put these things in for us. And I guess the other concern was whether you could actually hyperventilate to that degree and hold it there for 5 hours. And we had, as you may recall, these laboratory CO2 meters that record breath by breath expired entitled CO2. And we just basically had the same sort of thing. You put your meals on in the hospital, set on this tray all in front of the hospital bed, which was brought into the laboratory, and we just started hyperventilating and drove it down. And one of the other two of us would sit kind of cheering the person on. It was a long day. But we did it, and then when we got all through, I mean, Fred was kind of an enthusiast in all this, but he had, at the end of the day, a shaking chill. My suspicion is that he became a bit septic. Something was not completely sterile in the whole, I don't know, we were all very careful about this. And after it was all over, we'd had these 3 data points for all of these things. And then a little bit of extra data on Jerry Posner. Fred announced that we were all going to do it again. At that point, he was outvoted. Jerry Posner, I said, enough's enough. But it was actually published. I would think it was actually a pretty good study, but I don't think it'll ever be done again. I can't imagine doing that again, or asking anybody to do it.
Nor can I.
But it was a real, I mean, I felt like I had a first-hand introduction to measuring blood flow.
Well, Mark, I'm going to jump a little bit.
Sure.
I think it's fair to say that at least one of the reasons that you're sitting here this afternoon was your contributions made possible by PET.
Sure.
And I think it'd be important to hear from you the story of positron emission tomography.
How it happened.
How it got started.
Sure, sure.
The trials and tribulations.
Right. Well, the background to this is, of course, related very much to Michael Terpagosian, who, as you know, has been a faculty member of this institution for many, many years now. He came from France originally, and came to Washington University to get his PhD in the physics department. And while there, he became enamored with the idea that cyclotron-produced radiopharmaceuticals would be of interest in biology and medicine. And there was a cyclotron on the main campus, as you know. It's actually a very famous cyclotron, which has now been inactivated, but was put in there prior to the Second World War. And it was actually, as I understand the story, was financed by the Department of Radiology with the idea that it would produce radioisotopes that would be used in the radiology department for nuclear medicine.
Did that cyclotron have anything to do with the earlier presence of Arthur Holly Compton?
It may well have. I don't know his exact relationship to its purchase. But what did happen historically was that the Second World War came along, and it became part of the Manhattan Project. So it suddenly became a super secret research facility, and I think may have produced the first plutonium or something. I don't know what, but it played a very critical role. By the time the war was over, generator-produced isotopes that are now used routinely in nuclear medicine had taken over the field, and people didn't have much interest in this, except people like Mike Terpagosian, who thought that, you know, oxygen-15 and carbon-11 and fluorine-18 might have a role. So he did a number of studies in what was an old house trailer out beside the cyclotron building, in which they did a number of just basic pioneering observations sufficient to convince the National Institutes of Health to put a cyclotron in the Mallinckrodt Institute of Radiology. And he then became the physicist for the Mallinckrodt Institute and began to form a team of people to do research with this. A number of people have subsequently contributed significantly to this field. Anyway, after recruiting Michael Welch, the chemist, in about 1970, I was in a meeting in London, and he approached me and said, with your background in doing brain circulation metabolism, and wouldn't you like to come to St. Louis and work with these isotopes? And it just sounded enough different and interesting that I thought, yeah, that would be. But you have to realize, of course, that there was no CT, MR hadn't even been conceptualized, and what we were talking about was for using these isotopes, and in people anyway, injecting them up the carotid artery. And Terpagosian had developed a simple probe system that allowed, at that particular time, 6 areas of the human brain to be looked at as just big cones of the brain. But nevertheless, it was regional, and he and John Eichling, his graduate student, had figured out a way to measure oxygen metabolism. And this was the first time anybody had ever done any regional oxygen metabolism. And one of the carrots to induce me to come here was that they were going to build a system of probes over the head that would have 26 regions. And this was a big thing encased in lead and had all these things coming out the back. And in fact, it became christened as the chicken just because of its physical appearance. So I came here in 1971, and this thing was finally developed, and it was put down on the 3rd X-ray, and we were up and running, and we did a number of studies, even including some functional mapping studies, which have become a huge focus of what we do. But then Godfrey Hounsfield announced the development of the CT scanner. And I think that, well, I recall a good bit of this. The immediate focus of people like Terpagosian and Phelps and others here was, why, we can build a better CT scanner. So there was immediate interest in this, and people like Jerry Cox and Don Steiner quickly went to work and developed mathematical algorithms for the development of a CT scanner. And here it became very complicated. It actually, there was overtures from a company by, I think it was Picker, or one of the major radiological companies, and to develop some sort of working relationship to develop a CT scanner with people in the university working in conjunction with all of this. Well, this all kind of fell apart for reasons that are a little murky to me. And left people kind of disenchanted. And as I understand the sequence of events, Jerry Cox was, kind of wondering what in the year he had this wonderful algorithm that had been developed, and the question was what could be done about this. And the issue was raised, well, if we could reconstruct the density of the tissue by passing X-ray beams through, why couldn't we reconstruct the distribution of an isotope by looking at the emissions from the tissue? And it struck people like Cox and Phelps and others as a perfectly reasonable idea. There wasn't anything that would prevent this. Although there wasn't unanimous agreement within our group to proceed in this way, there was a considerable amount of disagreement about whether this should be pursued or it shouldn't. And in fact, the initial experiments were done at night by Michael Phelps to kind of provide the evidence that this was a viable idea. Anyway, it survived as a viable idea and became an infectious idea, as a matter of fact. And I became involved in that. We had christened this big chicken, if you will. And of course, to even begin to construct a PET scanner, you've got to have detectors and photomultiplier tubes and all of this. And the only supply of all this was this gadget that they'd built me. So the plea was, okay, we're gonna construct this thing, but we've got to take this gadget that you came to St. Louis to use apart. So they did. They tore it all apart, and the initial PET scanner was built on what virtually was a top of a table, in which they cut a hole in the middle of and laid these detectors around it and embedded them in lead and showed that, by gosh, you could reconstruct an image. And of course now the scanners themselves surround the subject and they turn and do all sorts of things, but at that point there was no mechanics and the objects in the center were turned, but it provided the evidence that this sort of thing would work. This was a remarkably, a number of people were involved in this. Including Terpagosian and Phelps and Snyder and Cox, and people over on the main campus, John Hood, and engineering people that began to think about how to put this all together as a physical entity. Mm-hmm. And that emerged. Amazingly, it emerged as what we called PET 3. PET 1 and PET 2 were just tabletop models. And so PET 3 actually emerged. And by gosh, the thing worked. Well, on our side of the coin, we had to begin to think, well, how are you going to make measurements with it? Because it's fine to have a piece of machinery, but what in the dickens are you going to measure with this thing? So we had to rethink a lot of the things that we had been doing. But this was an immensely exciting time. And once we had this machine, we began to think about how to do varying measurements with it. And you'd say, well, all then was going to be peace and tranquility and success. But in an environment as complicated as this one was, with all these very many people, and people kind of pushing several different frontiers at once, algorithm development, the physical development of these machines, the biology, and all of this was all kind of going on in parallel in a fairly small physical area in the Mallinckrodt Institute of Radiology. There were bound to be some bumps in the road. And the first one was the decision after this wonderful machine, PET 3, was up and going. The first decision, which had quite an impact on the group, was, well, heck, we've got a machine that's only a single ring. Everybody recognized that wasn't quite good enough. You ought to have a multi-ring machine. Let's build another machine. And the first mistake was how hard it was going to be to build machine number 2. So I went home, actually, one of the few years that I've gone back to Washington for Christmas rather than my parents and others coming here. And when I came back, Pet 3 had been sold. And I was told not to worry, that we'll have this new machine up and going in a matter of months. That, you know, this Pet 3, so off it went to Brookhaven National Laboratories, and Pet 4 was brought in. Well, Pet 4...
Mark, you mean, what was the carrot?
Well, the carrot was that we would have a multi-slice instrument rather than a single slice, that we could sample more of the brain. The carrot for the physicists was this was a new challenge.
No, but to sell the existing Pet 2, I guess it was.
Well, it was Pet 3. I think part of it was that they could use some of the money to build the next machine. But, I mean, in terms of today's machines, you're talking just pennies almost. I think Pet 3 was built for under $100,000 and was sold for $50,000. I mean, a modern-day PET scanner would be more in the range of $2 million. But anyway, off it went to Brookhaven, and it was 2.5 years later that we had a viable PET working again, because all of this development. And you'd think, well, you know, this could have thrown us off the track, but what it actually forced us to do, it was an agonizing period of time, because immediately the people at Brookhaven leaped into the PET world and were doing studies that we were incredibly envious of. But it did force us to think a lot about the way in which we were measuring things, and we spent a lot of time working with that. And I think in the long run it paid some big dividends. I think we would have taken approaches to things that at the time might have seemed very useful, but we probably wouldn't have explored things that we did, having to do particularly with measurements using oxygen-15. I think we virtually became the world's experts on using oxygen-15 radiopharmaceuticals at a time when others...
That was with the Pet 4?
No, we didn't even have a PET in there for a while. For 2.5 years, we didn't have a PET. When we had invented it and sent it off, it was 2.5 years. So we were using, you know, probes in animal studies and some studies on humans with injection up the carotid artery. But eventually we got first Pet 5, and then very shortly thereafter we got Pet 6, which arrived. And the time frame here, the first human PET study at Washington University was a study done in a patient with a stroke in April of 1973. I still have a slide of that. PET 6 was commissioned in 1980, so that there was some work done in between there, some very critical work, but the current era of PET imaging at Washington University then began for the brain. And PET 6 was unique, and it was the first instrument ever built strictly for the brain. And it was also unique in being driven by our desire to have an instrument that could measure quickly what we wanted to measure and could handle very, very high count rates, which was an immense advantage to us. It probably, in my estimation, is the best machine ever built here. It's still running today. We're using it for animal research now. But it began in work in 1980 and was decommissioned as a human instrument in 1993. So it went over 12 years worth of work and studied close to 3,000 subjects and amassed a tremendous amount of information on the human brain. Mm-hmm. And all of our techniques for, you know, mapping of function, all this were all developed on PET 6. So it was an incredibly productive time.
So, what's the current state of the art? I mean, what can we do now that we weren't able to do before?
Well, I mean, it's a lot of different things. We could, you know, all the things that I think people hope to be able to do, a whole range of measurements, biochemical and pharmacologic measurements on the human nervous system can be done really quite safely in patients and in normal people. And all of of that's been realized. You know, and it continues to expand. Some of it more slowly than others, depending upon the difficulty of the task and the problem that's involved. I think the other thing that came out of this was the seeds for which were planted well over 100 years ago was the notion that you could as well explore the function of the nervous system, and given the safety of the tools, that you could explore the function of the normal human nervous system. And actually going back into the 70s with some of the earlier equipment, we had tried these ideas out and had kind of plus-minus success. But when we got to PET-6 in about 1980, one of the very early things that was tried were things like opening and closing of a hand, or presenting a visual stimulus using light-emitting diodes in a set of goggles. And it was just immediately apparent that you could get tremendously powerful signals out of the brain by doing this.
Blood flow changes.
Yeah, these were changes in blood flow. And as I say, the idea that blood flow follows function was something that actually it's usually attributed to Charles Roy and Charles Sherrington in 1890. They published a seminal paper, but this idea had been talked about in the literature 10 to 15 years before that by a number of people, and in fact there had been some functional studies actually done on patients with head injury who had defects in their skull, and you could measure the pulsations in the skull. Some wonderful observations that in retrospect reflected this. But Roy and Sherrington kind of stated what the problem was and what the issues were. And then there was a long hiatus of things, and then John Fulton, working with Harvey Cushing, probably made the next most seminal observation. He had a patient was brought to Dr. Cushing who was losing his vision and had a large AV malformation in his occipital cortex. And among the patient's many complaints was the fact that he would hear this, he heard a bruit or a whooshing sound in the back of his head, particularly when he opened his eyes, and especially when he read. And John Fulton, who was ultimately to become one of the really preeminent physiologists in this country, was at that time working in the neurosurgical clinics with Harvey Cushing. And he took a great interest in this man. They operated upon him and apparently couldn't remove the AV malformation, but they paid attention to the man's history, and they could listen over the occipital cortex, and by gosh, they could hear this thing. And so they had the fellow read and close his eyes and open, and I mean, the sound increased when he read, and it went down when he didn't. And Fulton wrote this up in Brain. It's an absolutely wonderful description. It was written in 1928. But again, there was a hiatus until Seymour Ketty and all of his students, Lou Sokoloff and Bill Landau was working in that lab, kind of picked up the ball, and this was in the 40s, kind of right after the Second World War, and began to show again with techniques that were ultimately picked up and used by PET that there was a relationship between flow and function. And then of course all the Scandinavians, David Ingvar and Niels Lawson and all of these people picked this up, and this became an interesting thing. But I don't think it really reached the level of sophistication that it did when PET got involved in it, and not that PET itself was the key, although it provided a much better picture of it. But I think the other key aspect of this functional work was the wedding of the behavior to the technology of measuring these signals of brain activity. And of course you know that story with the McDonald Center, in that one of their charges, as you've explained to me, was that they bring some preeminent psychologist into this mix of things so that one could take advantage of the technology and really study brain in a serious way. And I think of all the people that were looked at, I don't think they could have, or you, could have found anybody better than Mike Posner. And the reason is that he represented an element of psychology which is known as cognitive psychology, which thinks of human performance in terms of information processing. The way they go about thinking about it is just ideally suited to wedding it to the imaging strategies that could be implemented on PET. And he spent a I think, an important 3 years here, and it was a mutually beneficial learning experience. It wasn't an immediate transfer of everything he did to Pet, because they don't do it quite that way, but he was flexible and he rolled up his sleeves and we did, and it was an immensely interesting time with a lot of young people contributing to the thinking process that brought all this about. But what we find ourselves positioned with at this point is a wonderful tool that allows us to think in a critical way about the general organization of cognition using these tools.
Mark, I think that's one of the most important aspects of what Pet can do. Look into this black box while we're thinking, and could you elaborate on that? I mean, you've already done some of these things that really are incredible.
Well, it does open up the black box, and I think the thing that's been particularly interesting, I mean, obviously people have developed ideas, whether it's in the neurosurgical suite or following patients with stroke. I mean, clearly we weren't completely ignorant about how the brain worked, but here you can get into this in absolutely normal people under relatively normal circumstances. Basically, they're just laying on their back in a scanner, so that presumably everything is operating normally. And so that I think that's an important perspective that up to this point had been lacking. And of course, it allows you, if you use a little ingenuity, to begin to approach processes having to do with language, which is such a uniquely human thing. And here you have a tool that can study the human brain, and to be able to go in there and begin to understand how it's organized and how it serves our processes of thinking and communicating and all that, I at least find very exciting. We've tackled issues related to attention and memory and
could you give some examples of that for posterity?
Yeah, oh gosh, yeah. Well, the language thing, you could just so I can give you a couple of examples of each of them. The language issue, of course, you could put somebody in a scanner and just have them, sing a song or recite the days of the week and this kind of thing. But that isn't very helpful in terms of trying to understand how areas are processing this kind of information. So the idea at the outset in the language area was to just make it a little simpler, and that is to deal with how do you deal with a word. So we've been dealing with single word processing now since, 6 or 8 years now, and we're still dealing with single words because there's a lot to learn. But we wanted, for example, to know what's different about looking at a word as compared to looking at, say, a dot on a screen. We typically present stimuli by presenting them, words on a television monitor that sits in front of the patient in the scanner. And so the first thing we wanted to know was that any different than just looking at the monitor itself with a little dot on it where you fix your eyes? And not surprisingly, it was dramatically different. If you open your eyes and look at a television monitor, you naturally see changes in the visual cortex. I mean, we weren't surprised that we did. But if you add to that screen words, you don't ask anybody to do anything, you just have them look at the screen, you get this remarkable elaboration of areas that are obviously in what we would call extrastriate visual areas that become active. Then it immediately raises a whole series of questions about, what are these areas and what is it about a word that is so interesting to the brain? And we've explored that a bit. You look into issues. Is it simply the fact that you've got a set of symbols there with vertical and horizontal lines, and it's just a very interesting visual stimulus with a lot of physical properties that stimulate a lot of areas of the system? Or is it related to the fact that you've got a string of letters, that somehow or other your nervous system is recognizing this? Or does it relate to the fact that that string of letters has some very interesting properties, not the least of which is that you can pronounce it? And finally, does it contain meaning? Well, you can construct then stimuli. In other words, you can construct things like false fonts, which look exactly like words and letters, but aren't words or letters, but have all the physical properties, and you can present them, or you can present consonant letter strings, which are strings of absolutely recognizable letters, but because they're all consonants, you can't pronounce them, and they don't have any meaning. Or you can present people with what we call pseudo words, which are combinations of vowels and consonants, and you can pronounce them, but they don't have any meaning. And you present words. So you step through this, and it turns out that, by gosh, the brain back there seems to have learned in the course of its development to recognize things that behave according to the rules of the English language. The thing that's turning on all these extra areas is largely pseudo words and words. So it allowed us to dissect this kind of thing out. But then you can step up a step further and say, well, okay, having looked at the words, now what happens if you speak what you see? So you subtract away the seeing and you speak, and it turns out at the outset that wasn't too surprising. I mean, the areas of the motor cortex and the supplementary motor area become active, and the cerebellum along the paramedian areas become active, and that all seemed fairly straightforward. I mean, it was nice to see, but it was very much predictable. And then we, in this same initial experiment, we were interested in the matter of how you get at the meaning of words. In other words, it may well be that if you're just asked to repeat a word you see, you may not even think a whole lot about what it means. On the other hand, we wanted to force you to do that. So within the group, they came up with a task which has occupied now years of our attention, and that was to take the same noun and ask you to generate a verb for it. So we'd show you the word hammer, and you'd say hit, or we'd show you knife, and you'd say cut, or car, you'd say drive. And if you subtract away the saying of the word, what you now add to the complex of areas, you kind of keep adding all these processing elements in there. You add left frontal and temporal cortex and the cingulate. And the big surprise was more of the cerebellum. It became a big player in this. Well, I think this kind of typifies the way we go about thinking about this. But and I guess if it had kind of laid out this way, you'd say, well, this is following more or less. The cerebellum was indeed a surprise. But one of the big surprises came quite by accident, as it does in science. And that is that it was realized that quite by accident, because a subject who was going to do this particular verb generation task on a particular occasion was actually rehearsed. There was concern that the subject wasn't too skilled at doing this, and they wanted to be sure that the subject could perform it. And examining the data from that subject, none of these areas, the frontal, the temporal, the cingulate, and the cerebellum, was there. There was no evidence of this. And this immediately alerted us to the possibility that a very brief process of learning could radically change the circuitry of the human brain. So we have pursued that with considerable interest, and indeed that's the case, that if you're confronted with an activity that is new and novel and demanding, it appears, at least in the language domain, that you're going to use a set of circuits initially that, once you're skilled at this task, are simply not used again. And so you use those to acquire these new skills, but then once you've acquired them, you pass them off to another. So it isn't just that this particular circuitry gets better at doing it. It has a particular job at the outset and then just is set aside.
Yeah.
And finding stuff like that has been really kind of neat. And we've, as I say, we've dealt with issues in attention memory. I'd be happy to talk about it, but I think this kind of exemplifies.
Gives us the flavor, right?
Yeah.
So, what about the future?
Well, I think one of the things we're faced with now, I mean, obviously, people will continue doing this kind of work, I think, for a long time to come, would be my suspicion. I think the thing we're faced with now is...
How about the future for Mark Raichle?
Oh, for me.
You've been involved with all of these studies, whether it was the psychiatrist looking for particular neurotransmitters, or the neurologist looking for blood flow, etc. Yeah.
Well,
You had a hand in all that.
Yeah, my focus, I must say, although it has been, you're right. I've been involved in pretty much every aspect of this at one point or another. But my major interest at this point is, I think, understanding a bit more about the normal human brain. I just find this a fascinating subject, and I guess one is particularly fascinated about it if you have some prospect of making some headway. And I, honest to gosh, think we do. Whether it's with PET or now as MR and other things are beginning to enter this, the tool is really kind of unimportant. I think the nifty thing is the question. And, you know, it's interesting that it's possible to, you know, contemplate issues, very important issues with regard to the nervous system, and it's even legitimate to utter the word consciousness. You know, that's been a kind of a philosophical concept for years, but I think neuroscience, people in the neurosciences, very serious neuroscientists, ultimately have now, you know, realized that the one thing that's truly, truly unique about the nervous system is that we're conscious. And the question is, how does that come about? How does that arise out of this thing? And, you know, I don't know whether I'll get an answer to that in my lifetime, but you can at least begin to think about the problem. I mean, even how you think about that. And it's embedded in the kind of stuff that we're doing. And I'm also interested in, you know, one of the small joys I've gotten out of this is that, because I still do some clinical neurology, I still attend. And the thing that I think has been particularly interesting is the impact that's had on my kind of clinical thinking about things. When I go to the bedside, I find myself now knowing a bit more about how the nervous system works normally, asking very different questions of a patient. And I find that just great fun to think, you know, if there's a problem here, I'm going to ask the question differently than I would have before I knew what I now know. And I would like to think that, you know, that will, if this continues to play out, make people who deal with neurological patients a little bit more insightful about what it is they're dealing with and what sort of problems their patients have.
Mark, what about Washington University? Its hand in all this story.
You know, it's been a absolutely critical in this whole thing for a variety of different reasons. Part of it, I mean, obviously what I do, here I am a neurologist, and I've resided in a department of radiology for 23 years with close ties to neurology and radiology and psychiatry and all of these other departments. And I think it's a true statement that there is an environment here that allows this kind of interdepartmental collaboration among groups to work as well as I think it could work anywhere. So that has been an important element of what I've done. I've obviously had some champions in all this, people that somehow or other had faith in what the devil I was doing and were willing to help when it was really important. I mean, one person clearly was Bill Landau. I remember thinking about, here I'd had this terror of a mentor, Fred Plum, and of course Bill Landau had an equally impressive reputation in the, but they're very different people. And Billy is a wonderful, wonderful advocate of what we wanted to do and was very supportive through a lot of this and of the people that wanted to work with me. And then of course, this was in terms of biological science, I'd have to say this is big science. It's costly and all of that. And I think the development of the McDonald Center through your efforts and Mr. McDonald's gift was critical. It's been absolutely critical on a couple of occasions. And then the kind of sustained support and backup that was needed, those things made the difference, I think, in the long run. So that the environment was all critical here, and having the right people around who were sympathetic and tolerant, because at times I think we might have been, you know, there was quite a crowd over there, and we were a bunch of kind of personalities, and we were probably not always the easiest to live with. Right. But I think the bottom line is I hope we've kind of repaid the university for what they've invested in it, because
Well, Mark, also the imaging center that's going up now, that is...
Well, this is kind of beyond anybody's wildest expectations. We've, of course, had this space. Now it seems terribly small and limited in the Mallinckrodt Institute of Radiology. Of course, they have put up with this, actually left us alone to do our own thing. And to that I can, in a way, thank Ron Evans. His major focus, of course, has been in the clinical arena, and they have a superb clinical training program. And we were just kind of left to our own devices to contend and do what we wanted to do. But now that the emphasis in radiology has shifted to a more balanced view of research and clinical work, we've been the recipient of increased support, and particularly in terms of space and facilities. And the Mallinckrodt Institute and the university is building now an imaging center, which is an absolutely, physically a phenomenal affair. It's going to be close to 70,000 square feet of space to house several major research groups that will collaborate in using PET and MR. If it isn't a success, it'll just be our own darn fault. It won't be because we didn't have the facilities to do this. I think it'll be a great sociological challenge to expand in personnel this rapidly and make it work. But I willingly accept a challenge like that.
Well, Mark, as usual, I always enjoy listening to you recount some history that was just current. And is there any other note that you want us to end on?
Really, I can't think of any. It's always fun to kind of chat about this. There's some kind of wonderful stories about all that's happened in this place.
Well, I want to thank you for letting us capture you for posterity.
You're welcome.